I'm here at TU Wien, and but most of the research I'm going to talk about has been achieved at École des Mines de Saint-Étienne. So I'm going to give this presentation with three main parts and some like kind of intermediate conclusions, and I would like you to like not hesitate to interrupt if you want to ask a question during the course of a presentation. So I'd like to show what we did in order to bridge continuum mechanics and biomedical applications. And first, I'm going to talk about how continuum mechanics models can predict human health. So this is more like something that this is more like chronological, and this is more like some of the first work I've done. And I'd like first to say that I mean, when we started to work in this field, and I started my group in Saint-Étienne, so I knew all the, I mean, computer modeling effort that was ongoing in different fields. But actually, when we looked at healthcare, then it was more like a trial and error approach when I mean, companies, they want to develop a new product or a new medical device. And so then we saw that there was like an ongoing effort from a number of institutions, and this started from this Physiomy project, and then after there was a creation of VPH, and now it's the VPH Institute, and we found a number of I mean, a number of like success stories, like HeartFlow, which is a company in the US that actually used continuum mechanics model that in order to predict, for instance, for HeartFlow, this is for the treatment of coronary stenosis. So this is a company in the US that has now I mean, a very high estimation at the stock exchange. So there were efforts also in Europe, in Belgium with FIOPS, but also in France, and France is quite active. And so I'd like to mention Zenzom and Abdul, but also in Montpellier, Simancure, or we also founded a startup in France, Predisearch. I'm going to explain a little bit the purpose of Predisearch. My own experience actually has always been based on establishing a collaboration with surgeons, with clinicians. And actually, what we were always interested in are aneurysms. So aneurysms, this is a disease that is mostly, so you can have also brain aneurysms, but we were always interested by aortic aneurysms. So the aorta is the artery that is here shown on this X-ray tomography, and it can balloon at different locations. So here, this is just after the heart. This is something we studied a lot, but it can also balloon in the abdominal part. And in the abdominal part, actually, what we found when we talked with surgeons is that it was very difficult for them to obtain medical devices that we call stent grafts. So this is what is used to have an intervention which will exclude the aneurysm from the blood circulation by making like something that takes the pressure, and it's made of a stent covered with a graft. And this is what is called an EVAR procedure, which means endovascular aortic repair. So endovascular because you do not open the body of a patient. And so for that, it was amazing, as I said at the beginning, how artisanal this procedure were planned. And so we started to propose to have finite element simulation of the device insertion and how the device would fit in the artery and how the artery would deform and make like the device not take the shape that it had when it was stress-free. And so this is what led us to this pre-research company, which is now providing a software to the companies making this EVAR device. And I'd like to mention like a collaboration with Marie-Laure Long, which is, nearby your institution. And they participated, for instance, to one of a clinical study where pre-research validated this, the software for this kind of device, which are the device with fenestration. And so the software needs to predict where we should have a fenestration in the device. And if you use this software now, you can gain like 2 or 3 months, which is very important due to the emergency of these interventions for the patients, the concerned patients. So this was just like more to have a kind of introduction about like the roots of our research. That there are computational models using continuum mechanics. They are used commonly now in healthcare, and there has been an evolution from what I found, I think, in 2008 and now, and the evolution has gone fast. But there are still major challenges. I would say that now the simulations which are used in the industries, they simulate medical devices, but what they fail still to, at least in the industry, what we do not find are models that could be used to establish digital twins integrating time evolutions. So what's going on? What's going to happen 1 year, 2 years, 3 years after implanting a device? So this is very challenging because now this needs not to take only into account like the mechanics of materials, but also the biology, which is going to have, complex coupling with mechanics. So I'm going to explain now some of the research we've done in order to address these challenges on a specific application, which is kind of very specific. We didn't find that we could have like a generic approach, but more like working specifically on one problem was already like, requiring a very important effort. So this is again on aneurysms, and so we show that we need to combine data-driven models to, the traditional continuum mechanics approach. So as I said, the challenge for these aortic aneurysms is decision-making. So the people having these aneurysms, the main risk they have is to have a dissection. So a dissection is that the wall of the artery has become weak when there is the aneurysm. And so the reason why there is an aneurysm is multifactorial. So it's very complex to say somebody will have an aneurysm or not. But it can be diagnosed. And when it's diagnosed, even in early stage, then it's very important to monitor, to have a surveillance of the patient because of this risk of dissection. But nobody knows yet why one patient has a higher risk of dissection. And this is where biomechanics has provided a lot of insight that now I think that there is a better understanding, and surgeons have improved the way they can surveil the patients. So one of the things that was discovered is that a lot of the patients having an aneurysm in the ascending thoracic aorta, there was also what I show here with these streamlines of the blood flow, and that here is shown with a schematic. There is a blood flow that we would say it's disturbed. So what is disturbed is more that usually the flow would be kind of parallel to the wall. But in some patients, and some of them are the ones who have like a bicuspid valve, which is a valve with 2 leaflets at the aortic valve, and normally it's a 3 leaflet. So it has been found that usually they have the flow that is, it makes a jet that impinges on the wall. And this creates a region here where there is the impingement. It has been found that it kind of induces reactions of the endothelial cells of the aorta that triggers some reactions in the tissue. And the main reaction is a proteolysis of a protein that is in the tissue. The name is elastin. And due to this proteolysis, then there is like, some damage, but there are cells that feel this damage and then repair the tissue. But by repairing the tissue, then it does something that in mechanics would look like this. So if you have the tissue at the beginning that would have this kind of stress strain behavior, if you just apply a uniaxial tensile test, if you consider this proteolytic injury that is induced by like the pathways that are that result from the disturbance I was talking about, then you will have this damage here. But then the reaction of the cells is going to induce a healing, but the healing will make like you have an offset, and this offset is the distension of the wall that creates the aneurysm. This is just because in order to induce the repair, there is need to remove fibers that were that has a too high tension, and this causes like the distension. So this is what we decided to model with a continuum mechanics approach in order to be able to understand what's behind this and how we could predict it. So what we used is something like continuum mechanicians are well familiar with. And also I saw that 1 month ago you had like one of the big shot of constraint mixture theory invited to your webinars with Gerard at Stephane. So just to say that here we're not using like all the coupling with chemo elasticity. Is just like a hyperelastic model that we use, and this hyperelastic model is a composite. Actually, we use like in the strain energy density function that we use for the wall. We consider like this is like a schematic of the wall. So there is a first layer here that is the media, and it's composed of smooth muscle cells, of collagen fibers, and of elastin. And we model it with a kind of averaging strain energy density function that contains the contribution of elastin, the contribution of collagen fibers with an exponential strain energy density, anisotropic, and also the contribution of the smooth muscle cells. And we have the same for the adventitia, which is the other layer, where we have different contributions. Especially we have much less elastin, almost no elastin, and we have mostly collagen and no smooth muscle cells. And why is it constrained? The thing is that actually, this is why you have always residual stresses in these soft tissues. The thing is that elastin here is produced kind of all at the beginning of the life of someone, and so the stretch it has is the stretch of the growth during like to go towards adulthood. So it's a large stretch. But the collagen here that we consider are fibers that are produced and removed regularly along the life. And so they keep a kind of always maintain the same stretch and the same stress actually. So this means that we have to consider that each component has a different reference configuration at which it has a stress-free state. And so this is why this is a constraint mixture theory model. And so all this after we have to load it and to induce in order to predict the equilibrium of the structure. So this is more like the passive behavior, but we consider growth and remodeling, which means that we have a biological effect. So the biological effects, they are modeled by the possibility of here changing the mass of each component. So this is the mass density. and these are like the strain energy densities per unit mass. So if we change the mass, then we change the mechanics, but also it just models that there is a production of collagen, for instance, here. So this is what we call growth. And remodeling is that what I said here, that this actually deposition stretch of collagen is going to evolve and to follow. If you have a growth of an aneurysm, then it's going to evolve and to follow the growth in order to keep the same tension of the collagen. So the change of mass is usually modeled like this, where actually you have this number that tells you here you have the stress in the fiber along the tension of the fiber. And here you have a homeostatic tension, which is the one that tends to be maintained in the tissue. And if there is a deviation between the two, then there's going to be production of new mass. And this is going to be scaled by a gain parameter that tells how fast the cells are able to produce this collagen. So this is what is in our model. And we can also have other reactions, like here you can also produce mass because you have an inflammation in the tissue, for instance. So in inflammation, you've heard what is an inflammation with COVID, I think, so I don't need to explain what it is. So this has been implemented using like this split between elastic and anelastic deformation gradients in the model. And so the anelastic deformation gradient takes into account this change of deposition stretch, and the elastic gradient is obtained through the equilibrium equations that are solved with this model. So with that, we modeled this effect I showed you before, which is that there is a region where there are these biological pathways that are triggered, and there is the destruction of the elastin protein. So this is the mass, the variation of the mass density of elastin. And then we simply model that at this location here, which then is local in space, and also in time, this occurs along a small time, then we have like a reduction, and then we predict the adaptation to that through the model I just showed before. So all this was implemented in two software, a commercial software, Abacus, and also an in-house software, because we had some troubles with using a different solver in Abacus. So we developed our own solver. And this is the kind of results that, that we were able to obtain. And one of the interesting things that we discovered, because actually these models were previously used to predict the growth of a theoretical aneurysm, idealized geometry. So if you have a cylinder and then it becomes like a balloon at the middle. That was the first time that we used this for patient-specific geometries. And what we found is that the insert that triggers the aneurysm was localized here. And this is where here, for instance, you have a maximum thickness change, because you have production of new collagen at this region to compensate for the elastin destruction. But the aneurysm growth is global. And the reason for that is that the homeostasis I was talking about, that the cells, they tend to maintain this, the collagen fibers always at the same tension. So if you disturb like the stress distribution here, then it redistributes the stress everywhere, and then this induces like a remodeling everywhere in the tissue. And this is why a local insult induces a global effect. So this was continuum mechanics, but we had to face a number of difficulties in order to go towards more like clinical application with this model, and we're still working on that. And I just wanted to show, for instance, what is, if you look at for instance, X-ray images, so CT images, then of a dissection, then you see here, this is a cross-section of the aorta, and this is like more like a side view with the dissection. So it's quite complex, and actually it would be very difficult to have like patient-specific boundary conditions to the model, but at the same time, we'll have a large sensitivity to these boundary conditions. So that's why we decided to kind of plug this model with statistical continuum mechanics model with statistical models by referring to some work. And so there was a European project. They published a very nice paper here that was related to heart mechanics, and they really kind of explained very well the combination of mechanistic approach with statistical models. And we found that actually our work so far was sitting mostly on that side, and we had to do the same as they did for the heart to couple it with that side as well. So that's what we started, and I'd like to give some of our achievements here. So we started with first setting up a cohort of patients with having a small aneurysms with a surgeon. So we have now 30 patients. They have like a regular meeting with a cardiologist, and every year we have them having an MRI scan. And so through this MRI scan, which is a 4D flow MRI, we can check what is the blood flow in their aorta, so that we can detect is varies locally this jet flow impingement. Okay, we had also a number of other patients. They went for the surgery, and then the surgeon, they removed the aneurysm, and we tested the aneurysm. And I remember that 7 years ago, I came to your lab, and I already gave a seminar, and this was mostly dedicated to explain what we were doing on this sample. So I guess that a lot of people are new in the lab, but I didn't want today to talk more about this. But what I wanted to say is that actually this brought us some statistical relationships between the properties that we get on this tissue and the local wall shear stress that we have from the blood flows on these patients, in order to have some relationships to apply to our model. Another thing was that we also developed for these patients we had like a tracking of the deformations of the aorta during a cardiac cycle. And with that, we could track, we could identify through an inverse approach the stiffness properties across the aorta, in order also to relate these stiffness properties to the initial conditions that we need to assign to our model, because the initial properties, they matter a lot on the outcome that we obtain for our growth and remodeling predictions. And the last thing that we did, and I mean, one of the papers has just been accepted, so this is still an ongoing effort. But what is really important, if you want to predict some kind of risk of dissection, you need also to have a rupture criterion. Rupture properties are probably like the most variable properties in the tissues. And so we thought that we should use artificial intelligence and machine learning, even deep learning, if we have enough data. And so we started to get into that field through a collaboration, because I'm not familiar at all with artificial intelligence. But we sent all our data to a group where they implemented an algorithm, and they found that they could learn actually where the fracture initiates in the test that we performed on the samples. And they found that if you use, for instance, a failure criterion based only on the stress distribution, then you would miss the location where a crack initiates. Like here it's missed, here it's missed. But with the AI approach, it's like a 95% success. And the main reason for that we found is that there is a strong correlation between the failure properties and the way that this exponential curve that we find on these tissues, the way it bends all along, because when it bends and it goes like a convex response, then it means that you have more and more collagen fibers contributing to the response. And the way it bends tells you how fast these collagen fibers they get to be involved. And this is something that actually can be related to a fracture. Last thing that we proposed is also to define some subgroups, and also in order to take into account variations that can be related to genetics. And this is going to bring me to the last part of my presentation. Here I'm going to explain this. So I showed you before like the growth of an aneurysm that is predicted by our model, but there is also an evolution of the material properties. And in our model, one of the things that I said that is important is that it always look at having the stress of the collagen fibers be, catch up with the change of the stresses that they receive, okay? And so the model always tries to take the collagen fiber again to homeostasis, being for us like the initial state. And so what we found is that this homeostasis is related to the contribution of the smooth muscle cells in the wall. And this has been demonstrated by considering animal models with genetic modifications. And it was found that they could not maintain the stress as it tries to be maintained in healthy individuals. So then we introduced in our model a parameter that is telling, what is the error that is made by the cells in like reading the stress in the tissue. And so if for instance, this is 1.1, then it will make like the collagen fibers have a 10% increase of stress because 10% of that stress is missed by the cells, and the same with 25% here. And this was one of the main result that brought us towards the last part of this presentation, which is dedicated to cell biomechanics, because we found that actually, if you have this parameter, then we do not see that the aneurysm grows much more than in that case here or here. It's almost the same shape at the end, but the obtained stiffness of the tissue is much bigger here than here. So we found that the same aneurysm would be obtained, but it would be much stiffer, so the tissue would be harder if we have, this missensing of the stress by the cells. And this is very important because all our previous research showed that the stiffness is a real risk factor. Like patients having a higher stiffness were the ones we had the higher risk of rupture. So this is why we decided that we should really investigate how the cells, they can apply or sense the stress of the tissue and maintain it in this homeostatic contribution. And so that's why we decided to go down to a scale and investigate the smooth muscle cells in the tissue and how they contribute to the mechanoregulation of aortic aneurysms. So I'm going to explain now this. I don't know, I said that I would stop and leave you ask questions, but I don't know if there are already questions, or if I continue until the end.
Don't worry, I think you can continue till the end and then I will ask questions. Thanks.
Okay. So, so as I said, so you have like, if this is a zoom here, a schematic zoom of this layer, the name is the media that I said that it contains smooth muscle cells and it contains elastin. So elastin, it's like plates, or we call lamellas, and you have like in the aorta of a human, you have about 60, and between each of these plates, you have the smooth muscle cells, and then in between, you have collagen fibers that like make some kind of connective tissue all around. And actually, it has been shown that if you have, for instance, here in the nucleus of these cells, if you have some genetic modifications, and one of them that is well known, has been studied a lot, is the Marfan syndrome. So this is a genetic modification to a gene that is responsible for the synthesis of a protein, fibrillin 1, then you will have an aneurysm. But there are like 20 or 25 of them that induce aneurysm in the people having these genetic modifications. So there is some kind of link between what's going on inside the nucleus of the cells and the regulation at the tissue level. So this is why we wanted to, okay, isolate these cells and look at what's different in their mechanics. So we're mechanicians. If they come from patients with aneurysms and patients who are healthy. So the first thing we started to do is called traction force microscopy. So the cells, so we learned how to culture these cells to get them from the tissue and that we receive from the patients and or from biobanks, and then to culture them on gels, and on gels which have fluorescent beds embedded, so that under a fluorescent microscope, we can have images of the deformations of the gel that we can obtain through digital image correlation, and then obtain the strains around the cells like this. So here you see a cell and you see strains around, or here, and then after, through inverse analysis, derive what's the force that the cell applies on the gel. So this is called traction force microscopy, because this is like a measurement of the traction forces of the cells. So usually people have used that a lot to study cancer cells, because they are very strong, and also this has a lot of implication, but not that much on smooth muscle cells. And cancer cells, they are more like a star shape with a number of points of tractions, but our smooth muscle cells, they are more like spindle shape with tractions mostly at the beats of the smooth muscle cell. So what we found, which was very interesting, so we tested about 1,000 cells. We had 3 donors, 3 aortic donors. So they were provided by a biobank, and almost same age, 3 aneurysm patients. So this is not a big number of patients, but this is still a first result that we found, that there was quite a stable value of the average of, this is not the average, this is the median, sorry, of the average of the median force of the smooth muscle cells coming from healthy patients. And there was a significant increase of traction forces of the aneurysm cells. And we even found after, but again, I say that we need to repeat this on other patients, that this patient that was lower actually is a patient who received all the aorta, and then we took all the aorta, we took the cells of all the aorta, so there are probably cells of the region where there is this jet impingement that I was talking about before, but there is also cells of the other side, and there is more like you see a big dispersion of the results for that one. So now what we're trying to do is to have a finite element model of the cell. So the cell has I don't, sorry. Yeah, so the cell, if you image a cell, what you see as major contributor to the structural response, you have what we call like the focal adhesion, is where there is a protein, the integrin, that can bind to the substrate. And you have stress fibers, and we model the stress fibers. We were inspired by a paper from the group of Abdul, and some of them, they cross the wool cell, and others, they are bound to to the nuclear envelope, and then we have the nucleus. And we modeled that, and we modeled like a contraction of stress fibers, because in the stress fibers you have two proteins, so they are made of actin, and the actin is a protein that polymerizes, and so this is this is the main constituent of the stress fibers. But on the actin, locally, you have another protein. This is the myosin, and the myosin is like a tightener. Okay, so it will bind, and then it can tighten, and it will tighten depending on, if I say it, I mean, in simple ways, on how much calcium it has available around. And so if you make like the concentration of calcium increase in the cell, then you can make it contract. Okay, but otherwise, then it would apply always like a basal or homeostatic tension. That is very important for us, because it's the one that will be by reaction applied on the tissue, and then that will induce the tension of collagen fibers I was talking about just before. So we had simulations that confirm that our model is kind of predicting well I mean, compared to the experiments, and also that the variations of the actin actomyosin I mean activation was making our results find the same dispersion as the experimental results, which was interesting for us, because there is such a big dispersion among the cells, but we could relate it mostly to the actomyosin activation, and we simply modeled it by a thermal activation model in here. This was developed in Abacus commercial software. So then what we did also is that on the same cells, we also look at their stiffness. For that, we used AFM indentation. So atomic force microscopy with the tip, we apply locally indentation, reaching a target force, and then we read the deflection for this target force, and then extract after an apparent stiffness parameter. And we were able, like for healthy cells or aneurysm cells, to map like stiffness in a region close to the focal adhesion, in order to understand if there were some differences between the cells, healthy cells or aneurysm cells. And we found that they were very similar. So here I show like elastic modulus obtained on these two cells, but then after we pulled like 20 cells, 20 healthy cells, 20 aneurysm cells, and we found that they always had like the median around 15 kPa, and no difference in between. So then the results that we came to is that actually, and that's something that we observed, is that the aneurysm cells, they tend to be bigger and wider than the smooth muscle cells from healthy patients, but the internal structure is not different. So it's a kind of hypertrophy that occurs in some of the cells, and we believe that it's because they need to apply stronger forces, larger forces, in order to repair the tissue. And we have an example for this. This is the myofibroblasts, which is another cell type. And these are the cells that will participate to healing if you have a scar. And just if you could cut your skin and then you have a scar, then after you will feel that the tissue is harder at the location of the healing. And we believe that this is this effect that we have in the aneurysm when it applies larger forces, because the tissue becomes stiffer. And we believe that these higher traction forces that we found are simply due because the cells, they become bigger in order to do their job of repairing the tissue. So that's where we are. So it's still ongoing, but we try now to get more towards the nucleus. And actually this year I'm here in TU Wien, and I'm working on developing a model for the nuclear envelope, and especially a protein that is in the nuclear envelope, and the name is the lamine. And lamine is very important. And there is a disease, the name is Progeria, and this is a disease very rare, less maybe than 1,000 people in the world. But at 10 years old, people, they look like 80 years old. And so this induces like a fast aging. And the reason why they die, so they die early, is always because of cardiovascular disease and aneurysms. So it means that if you have like defect, the protein that is located here, then you develop an aortic aneurysm, the same that we modeled before. So we try to have like a modeling of that nuclear envelope and try to induce all the cascade of reactions that would make our model grow the aneurysm in these patients and understand what are the links that induce that. So I'm coming close to the end now. I would like to say that, all these are like more like summary of the results that we have found, but it's still very important for us to understand like mechanoregulation even inside the cell. And so that's also something on which we are working. So among the projects we are working now, so this is to include that in an approach that leads to a digital twin. So the digital twin is not a simple virtual patient on which we can run some simulations, but it's also reading data from the patient. And so we try to have this approach, and this is through European project Meditate, which is an ITN network, and we try to follow things that were done, like I said, in cardiology, for instance. And also there is a nice paper from the American community where they explain also the integration of machine learning and continuum mechanics. And so we try to go towards this direction in this European project. And another thing that we'd like to do is to address, so I started my presentation explaining that with continuum mechanics, we could address the design of medical devices. But in the future, we'd like to address, again, using continuum mechanics, the design of drugs, okay? Because the drugs are going to target like the biological pathways, but this is going to induce reactions that will have mechanical impact. And so this is, for instance, what is explained in this paper, what about systems pharmacology. And here they found that by testing like 1,000 of existing drugs, they could find that some of them, by using a computational model, they could be repurposed to treat aortic aneurysms. So this kind of thing, I believe this would be very nice to, contribute to that with what we've developed so far. And with that, I close. So thank you all for listening. And I'm happy to take any questions. And I'd like to acknowledge the number of people who contributed to this work, and they were really always like very committed to their work, and also the funding that we received. Thank you.